In this video, we will talk about different types of synapses and spatial and temporal summation. A synapse is when a neuron communicates with another neuron or possibly a muscle or a gland. And so it is the area where the neuron and the effector or the neuron, other neuron, is going to meet. The first neuron in a synapse, if it's between two neurons, is called the presynaptic neuron. The second neuron is called the postsynaptic neuron because it comes after the synapse. The presynaptic neuron communicates via terminal boutons or synaptic knobs. They're also known as synaptic terminals, and they're going to impact a second neuron at its cell body or dendrites. And if it impacts enough, if it reaches threshold at the axon hillock, then it will cause an action potential in that second postsynaptic neuron. So this is showing you an image of a presynaptic neuron right here. And its terminal boutons or axon terminals are impacting or communicating with postsynaptic neurons. And an action potential will come down the axon and cause a change, which we'll discuss in this synaptic terminal. And this area right here is considered the synaptic cleft. There are two types of synapses. A chemical synapse is using chemicals, also known as neurotransmitters. An electrical synapse is a little different, and it's caused by transmission of voltage through channels called gap junctions, which means that these are cells that are right next to each other because they are connected via these gap junctions. So these are going to be cells that are going to propagate these are going to be cells that are going to propagate action potentials to cells right next to them. So these would be found in smooth and cardiac muscles. We're going to spend most of this time talking about chemical synapses. And so this is showing you an example of a chemical synapse. Here you see a presynaptic neuron up top and a postsynaptic neuron down below. And you can see an action potential is propagating down the length of the axon until it gets to the synaptic bulb. What's going to happen is they are going to open these voltage-gated calcium channels, which will cause calcium to rush into the synaptic terminal bulb. This calcium is going to bind to a sensor protein and make a protein complex, which is going to cause exfusion of the synaptic vesicles that are carrying neurotransmitter. They're going to fuse to the synaptic bulb and exocytose their neurotransmitters. And so we have neurotransmitter release in the synaptic cleft. Once neurotransmitters are released, we will have receptors on the postsynaptic cell that will bind and that can either excite or inhibit action in our postsynaptic cell. So depending on whether they will excite the postsynaptic neuron or inhibit the postsynaptic neuron, they are called EPSPs or IPSPs. EPSP stands for excitatory postsynaptic potential, and it is called excitatory because it's going to move the membrane potential of the postsynaptic neuron closer to positive. So it's going to depolarize it, which it's moving it closer to threshold. So that means that it's going to make it easier for an action potential to occur. An inhibitory postsynaptic potential is going to do the opposite of that. It's going to move the membrane potential further away from the threshold, so it's going to make it harder for an action potential to occur. This can happen by opening chloride channels or potassium channels. If it opens chloride channels, chloride will move in, making it more negative. If it opens potassium channels, potassium will move out, also making it more negative. Remember that this means that it's hyperpolarization, so it'll make it more challenging for threshold to be reached, and that's why it's called inhibitory. So we say that EPSPs and IPSPs are graded, and what that means is that they actually don't occur in our axon. They occur, as the images showed you before, in the dendrites and the cell body of our postsynaptic neuron. And this is considered graded because one EPSP is not going to be able to cause an action potential. It has to be several EPSPs acting together, adding together to produce enough of a depolarization to reach threshold. And this is why it's called graded, because it's kind of adding up together. So if it reaches threshold right here at the axon hillock, and then if that occurs, then that can lead to an action potential. So as this picture shows, there are a few EPSPs occurring. This one only brings it up to, negative 65 maybe. This one brings it up to negative 60. And then a third one possibly can bring it up to threshold. And if that threshold occurs right here, it is going to cause that action potential to propagate down the axon. So I said that IPSPs are included in this too. However, IPSPs are going to counter what EPSPs do. And so if there are EPSPs occurring and IPSPs occurring at the same time, they're going to cancel each other out. And so if there's one IPSP from one neuron plus an EPSP from another neuron, they're going to completely cancel each other out and nothing is going to happen. So what we're saying here is that our EPSPs and IPSPs that occur in the dendrites and the cell body, they are going to summate. They are going to add together. Because remember, only one EPSP will not do it. It has to be several EPSPs occurring at the same time to reach threshold at the axon hillock for this to occur. So there are two types of summation. The first type is called temporal summation, and these are EPSPs that are occurring from the same neuron, so from the same presynaptic neuron. They're going to occur closely in time, because if one occurs in time and then, it waits and then another one occurs in time, these are not going to summate. But if they occur once in time and then quickly again, then it will summate. And so temporal stands for time. They have to occur closely in time before they fade away. And, EPSPs and IPSPs do not have a refractory period. These are not our action potentials that we were talking about last week. This means that they are going to be able to summate, unlike our action potentials. If enough summate closely in time, then threshold can reach, and then an action potential can occur. So that's temporal summation. The second type is called spatial summation, and these are EPSPs from different synaptic neurons, presynaptic neurons. But they occur all at the same time, and they're all very close to each other. And so if they occur all at the same time, they're really close, they are able to summate, and then threshold can reach. So here is an example of spatial summation. This first example shows release of a neurotransmitter from neuron 1 only. So here is an example. This shows release of neurotransmitter from only one neuron with an EPSP. It gives it a little depolarization bump. But this example over here shows release of neurotransmitter from two neurons occurring pretty closely in time together or at the same time. And as you can see, they will summate, they will reach threshold, and an action potential will occur. So let's review what occurs in the presynaptic neuron when an action potential propagates down the axon toward the axon terminal. The action potential is going to come down. As it comes down towards that terminal bouton, it will open voltage-gated calcium channels, and that will cause the fusion of vesicles and release of neurotransmitter into the synaptic cleft. That will get bound to our postsynaptic neuron, and that binding is going to cause chemically gated channels to open, and this will cause inward diffusion of sodium. So sodium will rush into the cell. So this is small, this is localized, this is not your action potential that we talked about last week. This is just a small change, and it might cause a small depolarization bump, right? Well, so that's why it's called an EPSP. If there is summation and there are enough EPSPs that threshold is reached at the axon hillock, that is going to cause what we discussed last week with open voltage-gated sodium channels to occur, and then potassium channels to occur, which will be our conduction of an action potential down the length of an axon. So that's kind of putting together what we were discussing today with what we've discussed last week. So excitatory postsynaptic potentials depolarize the membrane because they are able to open these voltage-gated sodium channels that are around the cell body or the dendrites. No, again, this is not what we talked about last week. This is different, this is graded, this is local, and it only occurs in this area. So it's graded in magnitude. It has no threshold. It has no refractory period. It's not like there's a time when one of these graded potentials can't occur. And these add together. This is not true of our action potentials. Remember, our action potentials cannot gain in amplification. So as long as these can add together and reach threshold at the axon hillock, then there will be an action potential. Cable properties. This can cause our EPSPs to fade over time and distance as well because we have leakage of positive ions. So again, that is why we need more than one EPSP to reach threshold, and we need them to work closely in time and in space. These neurotransmitters are going to be able to activate different types of channels, and so these are chemically regulated channels. First one is called a ligand-gated channel, and pretty much what that means is that a neurotransmitter is going to be bound to it, and it's going to open up an ion channel. That's kind of the ones that we've been discussing with our sodium or potassium voltage-gated channels. So when a ligand, which just means a molecule that binds to something, so a neurotransmitter is a ligand. When a ligand binds to the receptor, it's going to open a channel. These are also known as nicotinic receptors for the neurotransmitter acetylcholine. And then there are G protein coupled channels, and we spoke about this before, I believe. Neurotransmitters are going to bind here, and it's going to cause activation of these G proteins. Remember, they were alpha, beta, and gamma. These, for the neurotransmitter acetylcholine, these are called muscarinic receptors. So this is review, hopefully, but G proteins are alpha, beta, gamma. They are going to dissociate and they can activate an effector. So we've only been talking about acetylcholine here. Nicotinic and muscarinic receptors only have to do with acetylcholine, but acetylcholine is a really important neurotransmitter, and maybe you learned this in anatomy. It's used in the brain, it's used in the autonomic nervous system. It is what activates our skeletal muscle. That's what we find in our neuromuscular junctions. And so nicotinic acetylcholine receptors that are going to open up an ion channel, we find those in our skeletal muscle. So they activate our muscle contraction. That's one really important place we find that. Muscarinic acetylcholine receptors, which activate those G proteins, we find those in cardiac muscle and smooth muscle, so within those gap junctions. And so an example of what they can do is to make your heart rate decrease.